Welcome to week 15. Today I'm going to be discussing pathogens of the digestive system or the gastrointestinal tract. And while I'm going over today's lecture, I also want to focus a lot on reviewing material that relates to what we've learned in units 1 through 3. On your exam next week, we're going to have at least 50 questions that relate to unit 4, and then we'll have about 25 questions that are review, but they'll be in relation to what you're learning in this unit. So as we go through the lecture, if something pops up that is worthy of reviewing, we're going to take a little extra time to do that. All right, the overall structure of today's lecture is as usual for this unit. We're going to cover the anatomy and defenses of the GI tract and then go over 5 different pathogens for today. All right, so infections can occur nearly all along the GI tract. So the gastrointestinal tract is also called the digestive system. Gastrointestinal tract refers to the anatomical regions. Digestive system refers to the function, which is to digest our food. And the food that we eat gets ingested into the oral cavity. And our food that we eat is made up of our 4 classes of macromolecules. So you should remember what those are. We have carbohydrates, lipids, proteins, and nucleic acids. And they get broken down into the small monomer subunits. Or components. So for example, fats are made up of glycerol and fatty acids. So they're not broken down into monomers, they're broken down into that glycerol compound and individual fatty acids. All right, so we eat food in the oral cavity. As we swallow food, it moves down into the esophagus and into the stomach. In our stomachs, digestion starts occurring of proteins. Carbohydrate digestion begins in the mouth, so some starches are broken down in our mouth, proteins are broken down in the stomach, and then in the small intestine, especially where the small intestine meets up with the pancreas and the liver, all of this digestion happens right here in the small intestine. In the latter portions of the small intestine, we're going to have absorption of the nutrients. So we break down our macromolecules into our smaller monomer subunits, and then those smaller nutrients are absorbed into our bloodstream. When the undigested food then enters the large intestine, this portion right here that's made up of an ascending, transverse, and descending colon, these portions of the large intestine are packed with microorganisms, just eating away at the undigested materials. And then water is absorbed to form back into the bloodstream, and so feces is formed and excreted through the anus from the rectum. So if there are microbes all along the digestive tract, from the mouth to the stomach to the small intestine to the large intestine, microbes populate every single portion of this. And so every place is a potential site for infection. In the oral cavity, if we have growth of fermenting and anaerobic microbes along the gum line or in our teeth, we can get infections like gingivitis or dental cavities. The esophagus can become inflamed. Oftentimes that's due to acid reflux from the stomach. But if the stomach gets inflamed or infected, we call that gastritis. So gastric or gastritis, if you see these words, it'll refer to the stomach. Anything that in turn refers to the intestines will be referred to as enteric. So the small intestines is where our digestion and nutrient absorption occurs. The large intestine is where primarily just water absorption occurs, and microbes are chomping away at the rest of our undigested food. So if an infection is called gastroenteritis, that means it can infect all portions of the stomach to the small intestine to the large intestine. The organs that are attached to the gastrointestinal tract include the salivary glands. Very few infections occur there. The pancreas, if that gets infected, it'd be called pancreatitis. And if the liver gets infected, that would be called hepatitis. So hepatic usually refers to the liver. Okay, so the gastrointestinal tract is packed with a number of natural defenses because of there being so many microbes entering and leaving on a daily basis. The first two have to do with a physical flushing barrier. All of the cells lining the epithelia secrete mucus. So there are constant mucus secretions preventing attachment to our epithelial lining. And then peristalsis is the muscle movement. So this keeps fluid and microbes moving in one direction, and that helps to prevent attachment. Of course, we have a number of good microbes or normal microbes that inhabit the gastrointestinal tract. And so those normal microbes would be a physical or a biological barrier to infection. Lysozyme, this is an enzyme that's found in our saliva. Just to review what lysozyme is, it is an enzyme that digests peptidoglycan. Okay, so a good review question that could follow this would be, where do we find peptidoglycan? Do we find it in the cell membrane of all cells, the cell membrane of only bacteria, the cell wall of all cells, or the cell wall of bacteria only? And that would be the last one. Peptidoglycan is found only in the cell walls of just bacteria. Okay. The stomach has a very highly acidic pH, so many microorganisms cannot pass through the stomach. So the stomach isn't sterile, but many of the microbes are killed as they pass through the stomach. The stomach has a pH of 2, so that's very acidic, and highly acidic environments have a high proton concentration. So remember that as pH goes down or acidity increases, the proton concentration of a solution increases. So pH and hydrogen ion concentration are inversely proportional. All right, when the food from the stomach goes into the small intestine, bile is secreted into the small intestine from the liver, or from the gallbladder, in fact, which stores the bile. Bile itself, or bile salts, have an antimicrobial property. And so many organisms or enteric species are inhibited by bile salts. In fact, you have used bile salts in some of your selective media in the lab to help identify different types of the gram-positive cocci. And then there's lymphatic tissue surrounding the gut, called gut-associated lymphatic tissue, which is called GALT. All right, so I mentioned that microbes inhabit the gastrointestinal tract all along the GI tract from the mouth to the anus. I'd like to present a little bit of a diagram on how this relates to oxygen levels. So, as you can imagine, we breathe in air, So oxygen is going to be a very high concentration in the mouth. As we swallow our food and it goes into the stomach, it's going to be a little lower in the small stomach into the small intestine. And then towards the end of the small intestine, all of that oxygen is going to be depleted. And there's going to be absolutely no oxygen from the end of the small intestine to the large intestine. And so in this small and large intestinal area, we're going to have our anaerobes. So our anaerobes include facultative anaerobes that can grow facultative. These are organisms that can grow with or without oxygen. If oxygen's present, it'll consume it really quick. And this leaves an anaerobic environment for our obligate or strict anaerobes to grow. All right, so how do these organisms make a living? They take the nutrients that we don't digest and they use them for energy by various forms of glucose catabolism or related energy forming metabolism types. So they can grow by anaerobic respiration, where an alternate electron acceptor is used in that electron transport chain. Or they can grow by fermentation, where organic compounds are broken down partially, and then they have to be reduced again to recycle that NADH back into NAD+. So make sure you review the general differences between aerobic respiration, which would occur by many microbes in our oral cavity, where there's a lot of oxygen, and anaerobic respiration versus fermentation. So you have an old summary chart that you can look at and that long diagram that we drew on the board as well. Okay, so when we hit the stomach, there's going to be fewer microbes. I'm not sure of the exact amount, but let's say about 10 to the third microbes. And then in the small intestine, microbes start growing again. They become about 1 million microbes per milliliter in the parts of the small intestine that are closer to the stomach. And then further on down the small intestine, they increase by another 100-fold. And then by the large intestine, they increase in number by 1,000-fold. In the GI tract total, there are 10 to the 14 microbes total. And that is equivalent to 100 trillion microbes. In fact, the human body itself is made up of between 10 and 100 trillion cells. The reference I have below says 10 to the 13th, which is 10 trillion. A more recent reference that I've found suggests it is closer to 100 trillion. But if you take this range right here and compare it to how many microbes are found in the GI tract, you will see that our GI tract contains just as many microbial cells as we have human cells in our entire body. So are we more microbe than human? You'll have to decide for yourself. Okay, we're going to get into the pathogens now. We're going to start in the stomach and work our way down to the intestines. Our first pathogen is a gram-negative bacterium called Helicobacter pylori. So let's first review our flow chart of microorganisms. We've got all cells versus viruses. And then within our cells, we have prokaryotic and eukaryotic cells. And the prokaryotic cells are those that do not have a nucleus, and all of the organisms are single-celled. We've got our bacteria, and we have our archaea, which are 2 of the 3 domains, the third domain being eukarya. And in our bacteria, we've got gram-positive bacteria, and we have gram-negative bacteria, and then a third category called acid-fast bacteria. I'm going to leave that here for now. So gram-negatives have all the properties of bacteria, which have all the properties of prokaryotes, which have all the properties of cells. And then how gram-negative bacteria are different from gram-positive are that they have a thin peptidoglycan cell wall, whereas gram-positive bacteria have a very thick peptidoglycan cell wall. And they have an outer membrane that contains lipoproteins, lipopolysaccharides, which is endotoxin, and porins. I'm going to just leave this one up here since that's one of the 3 virulence factors that you need to know about. So because it's a gram-negative pathogen, that means it has lipopolysaccharide by definition of what gram-negative means. It's also a flagellate, and the cell shape or the cell morphology is that it is a sporillum. Not a spirochete, but just a basic sporillum. And do you all remember what it is called when a bacterium has flagella at the end of the cell, but there's many of them? Is it monotrichous, paratrichous, amphitrichous, or lophotrichous? Correct. It's lophotrichous. Good job. All right. And then a sporillum just means it's a spiral-shaped cell. The 2 infections caused by Helicobacter pylori are both upper digestive or upper GI tract infections. The stomach is part of the upper gastrointestinal tract. So these are upper GI. That includes the mouth, esophagus, and stomach. Specifically, gastritis is in the stomach. Same thing with gastric ulcers. Gastritis is the more mild form of the infection. It's an inflammation and infection of the stomach lining. Symptoms include burning pains emanating from the abdominal regions, whereas an ulcer is a lesion in the lining, and you'll have the same symptoms as above, but also potentially bloody vomit or bloody stools. And the story of how Helicobacter pylori causes this infection is very interesting. So there were a pair of scientists, Barry Marshall and Robin Warren. They were working together on a hypothesis that Helicobacter pylori was the cause of gastric ulcers. It was previously thought that gastritis and gastric ulcers was caused by environmental conditions like spicy food or a high sugar diet or stress. Now we know that these conditions can just trigger or aggravate the bacterium into causing an infection. And so how this was determined is they were working, they had found in biopsies of a number of patients that there was this H. pylori present. But that wasn't enough to say that the H. pylori caused the infection. Because if you recall, in Koch's postulates, not only do you identify the pathogen from infected individuals, that's just the first step. After you isolate the pathogen from infected species, you then have to get it in pure culture, which they did, and then you inoculate an animal model and make sure it causes the same disease. And then you look for the disease again. The disease should present itself. And then you re-isolate the organism. All right, so they knew, Barry Marshall and Robin Warren, they knew that H. pylori was isolated from the biopsies of people with gastritis and gastric ulcers. They got it into pure culture, but they couldn't get it to infect an animal model. And this is why animal models aren't perfect, because other animals might have different host cell receptors, or they might not be infected in the same way as humans. So what Barry Marshall did is he drank the broth culture himself. He infected his own stomach. And 5 days later, symptoms appeared. 8 days later, he got a biopsy. And sure enough, from that biopsy, they did the tissue work and the microscopy and the culture work and found that the Helicobacter was associated with that disease. So this pair of scientists did Koch's postulates on one of themselves. And they were published, and they have since received a Nobel Prize for their work. It is not allowed that we do this in science today. Different time back then. So Helicobacter pylori, I'm going to go back up for a minute. I have mentioned that it's a normal stomach microbe, or I put that in my notes. It's actually found in 2 out of 3 human beings in the world. So, and in fact, if you don't have Helicobacter pylori, it's associated with a higher likelihood of autoimmune diseases or a lowered immune response. So Helicobacter pylori is not just a normal stomach microbe, but it's also considered a good microbe most of the time. And it's not fully understood, how do good microbes go bad? But we do know by looking at the genome of Helicobacter pylori and studying it in pure culture that it does have virulence factors that help it survive in the stomach and cause disease. So one virulence factor that helps it survive in the stomach environment is an enzyme called urease. And this is one of the enzymes you tested in your gram-negative Enterobacteriaceae tests. And so what it does to help survive in this acidic stomach is it breaks down a compound called urea, which is naturally found in our blood. It gets exported in the kidneys, in urine, and excreted out of the body, but there's always a small bit of urea in our bloodstream. And it breaks urea down into both ammonia, which is a base, and bicarbonate, which is a buffer. So this helps neutralize the pH of its direct surroundings, allowing it to be able to grow under such harsh conditions. So the combination of these two things helps it neutralize that acidity in its direct surroundings. Because most of us have Helicobacter pylori, there's no way to know how it's transmitted. It's endogenous, it's a part of us already. What triggers infections is still not extremely well understood, but stress, anything that weakens the immune system, will likely have something to do with it. Because it's a bacterial infection, antibiotics can be used to treat gastric ulcers. And because many of us have it, there's no known way to prevent gastritis infections. All right, we're going to move down into the lower gastrointestinal tract now. The lower GI tract includes the small and the large intestines. Many infections of the lower GI tract will be called gastroenteritis for infection of the stomach and small intestines, or enteritis, or just diarrhea. All right. C. diff, Clostridium is the genus name, difficile is the species name, and it is difficult to get rid of because it forms endospores. So remember that endospores are found in 2 genuses. One we work with a lot in lab, Bacillus. Bacillus is not just a cell shape, but it's also a genus name. So Bacillus cereus, Bacillus anthracis, these would be pathogens that form endospores. And then, of course, also all of the species within the genus Clostridium. All right, so C. diff, for short, is gram-positive, so it has that thick peptidoglycan cell wall with teichoic acids and lipoteichoic acids holding it together. It's bacilli, it's in its cell morphology, which means it's rod-shaped, and it is an endospore-forming bacterium, And so after these vegetative cells die, the endospore can live for a long period of time, which makes it very hard to get rid of. It's very tough and resistant to killing. Remember, in order to kill endospores, you need sterilants in order to kill them. So you need some sort of method that can be used for sterilization, like the autoclave, steam heat under pressure, maybe bleach, hydrogen peroxide, something that will kill all microbes, not just most microbes. So sterilants are effective in killing endospores. All right. C. diff is a common infection that's acquired in hospitals. So we call them HAIs for hospital-acquired infections. And the infection it specifically causes is, well, your book calls it a C. diff infection. So C for Clostridium, D for difficile, and I for infection. So obviously a C. diff infection is caused by Clostridium difficile, but it's also called antibiotic-associated diarrhea. And that is because when a patient in a hospital takes antibiotics, its normal microbiome is completely wiped out. And C. diff takes the opportunity to then colonize that wiped out GI tract and cause infection. So it's also an opportunistic pathogen. After antibiotics wipe out the normal microbiome, Clostridium difficile takes advantage of that, colonizes, causes infection. There are 14,000 deaths per year attributed to C. diff infections. The CDC considers it an urgent threat. And even if infections are treated, they're often recurring. About 20% of individuals that have come down with a C. diff infection will get that infection again. So infections can be recurring in about 20% of the cases. Luckily, there are alternative therapies that have a much higher success rate than antibiotic treatment of C. diff infections. And those alternatives are not antibiotics, but rather probiotics. And in fact, it's not supplements that are usually given. In fact, it is full, I don't want to say blown, that sounds not appropriate in this instance, but full fecal transplants. So taking the fecal matter out of one individual and replacing it with the stool from a healthy individual. You can actually get paid to be a fecal donor in some areas if you have a healthy stool. Something to look into. All right, our third pathogen on the list is Escherichia coli or E. coli 0157H7. This isn't just regular E. coli, so this is not our normal good E. coli. E. coli is a normal part of our gastrointestinal tract, and this E. coli, so let's say here's our good E. coli. That's supposed to be a smiley face. And E. coli 0157H7 is bad E. coli. All right. It is gram-negative. It is a member of the Enterobacteriaceae family. So other members of that family include Klebsiella, Proteus, and Citrobacter, Campylobacter. It causes enterohemorrhagic diarrhea or bloody diarrhea. Entero means the intestines, hemorrhagic means bleeding. And so it causes an acute or a severe gastroenteritis or diarrhea that is accompanied by bloody stool. It can cause such severe diarrhea that dehydration can be a cause of death, especially in young children. But what is also a cause of death is a complication called hemolytic uremic syndrome. This is a type of anemia that results from this EHEC infection, and it results in kidney damage and also kidney failure. So it is associated with an incredibly low infectious dose. Only 10 cells are needed to cause an infection. That means E. coli 0157H7 can outcompete other microbes and attach with extreme efficiency. If only 10 cells are ingested, enough will get through the stomach into the small intestine, be able to attach, start dividing, and cause the infection. So this is really low, so that means if there is an outbreak, just a little bit of that microbe will make you sick. So it's very dangerous having such a low infectious dose. The toxin that results in bleeding is called Shiga toxin. So I've got a fun story about that and how it was discovered. Shiga toxin comes from the genus Shigella, which is also part of the Enterobacteriaceae family. Shigella is a GI tract pathogen. And it creates has the gene for Shiga toxin. So how did Shiga toxin get into E. coli and make E. coli a pathogen? It was found that a virus picked up this gene, so a bacteriophage. So a phage infects Shigella and then goes through the lytic cycle and produces more phage. And one of these phage was carrying the Shiga toxin gene. And this phage then infected an E. coli cell, and E. coli took up the viral genes, including the Shiga toxin gene. And the way they know this is that when they did the genome sequencing, they found a Shiga toxin gene surrounded by viral genes. And that Shiga toxin gene was identical to the one found in Shigella. So scientists knew that a virus must have picked up that gene from Shigella and transferred it into E. coli. And what do we call this type of gene transfer? Where a virus is responsible for transferring a gene from one organism to another. We call that type of gene transfer transduction. So that relates to Unit 2, genetic processes. All right, so E. coli 0157H7, or EHEC. It is always transmitted through the fecal-oral route of transmission. What that means is some E. coli comes out of the GI tract of one animal, and it ends up in the mouths of humans. So from feces to oral. It doesn't always have to come from human. It can come from other warm-blooded animals where E. coli can infect. So cattle are also are carriers for E. coli 0157H7. Not just the cattle, but manure that's spread on farm soil. So most of the outbreaks that are associated with E. coli 0157H7 are from ground beef. But produce is also a very common source or vehicle. So a vehicle is contaminated meat or produce. Vehicles are primarily responsible for the transmission of these infections, and produce and ground beef are the #1 vehicles. Fomites can also be involved in an indirect transfer, but that's less common than these vehicles. So cooking your meat thoroughly is what can prevent these. So especially think about ground beef, right? If you think of a steak, you can have a steak medium rare because when you cook the outside, all of the microbes sizzle and die, right? Heat will kill all of the microbes on the surface of the steak, and the inside of the meat, because it's muscle tissue, is sterile. But when you have ground beef, right, you take a piece of meat where microbes are only on the outside and the inside is sterile, but then you grind and mix it all up. So in ground beef, microbes are all the way through. So if you're eating a hamburger, you want to make sure you cook it thoroughly all the way into the middle. Right. And then washing produce can also help, but if there's produce known to be infected with that came from farm soil that was contaminated with E. coli because manure that was applied on that soil, then oftentimes washing produce won't help because the E. coli might be inside the produce rather than on the surface of it. Antibiotics theoretically work to treat E. coli infections, but they're very risky. The reason is, if the infection is severe, you have a whole bunch of E. coli built up. And remember, E. coli is gram negative. And so because it's gram negative, it has endotoxin, right, which is a part of its outer membrane. So if you take antibiotics, sure, the bacteria will die, but it might result in a greater risk of that endotoxin getting in your bloodstream. It might result in a higher risk of that complication called the hemolytic uremic syndrome, and it might be more likely to result in a deadly infection. So oftentimes, doctors will let the infection pass with our normal immune system trying to take care of it and using fluids to prevent the dehydration associated with that diarrhea. All right, we've talked about two different cases of diarrhea. We've talked about C. diff and we've talked about EHEC. Now let's talk about viral gastroenteritis. The virus associated with outbreaks, for example, cruise ship outbreaks, is norovirus. So norovirus is, it's not often deadly, but it will result in an infection lasting for a few days. All right, so the symptoms associated with gastroenteritis are diarrhea, vomiting, nausea, cramping, etc. So all along the GI tract, you'll get symptoms. Norovirus is one of two major causes of viral gastroenteritis. The other is rotavirus, but as you see here, there's a vaccine for rotavirus, and it also doesn't have as low of an infectious dose, so it's not as associated with the outbreaks like norovirus is. So norovirus is a single-stranded RNA virus. So remember, every virus has a protein capsid and a genome inside, and norovirus happens to be single-stranded RNA on the inside. It does not have an envelope, and the incubation period is anywhere from half of a day to 2 days. It has a very low infectious dose, which is why outbreaks can be very common, especially where everyone's in closed quarters like a cruise ship, and just a small number of that virus can make somebody else sick. There's also no vaccine, yet another reason for epidemics being common. Our final cause of diarrhea is going to be protozoan. We have Giardia lamblia. Giardia is a protozoan. So remember, on our flow chart of cells versus viruses, we've got our prokaryotic cells and we've got our eukaryotic cells. And protozoa, despite starting with pro, is one of our types of eukaryotes. So in our eukaryotes, we have fungi and we have protozoa. And we have 4 different types of protozoa depending on their motility. This particular type of protozoa, Giardia lamblia, is a flagellate. So you can see the flagella all around the cell. Giardia comes in two forms. The trophozoite form is the flagellated form, and this is the form that causes infection in our small intestines. The cyst form is a dormant state, similar to a bacterial endospore in function, but different in structure. It's not as tough to kill, but a cyst is just a dormant structure of protozoa. We have cysts that are found, we saw that with toxoplasma, that can survive for a long period of time in cat litter boxes. Cysts can also last for a long period of time in recreational and other types of natural water supplies. So the cyst form is the dormant form, and this is the form that can be ingested from water. If you ingest cysts from unclean or untreated water, then in your GI tract, its life cycle will go back to the flagellate form and it'll cause an infection. Right, so it's got a life cycle including both of these forms. How does it get in the water? It doesn't really get in the water from humans. It gets in the water from other animals. So it is a zoonotic illness. The cysts can come from the fecal material of beavers, from cattle, rodents, and sheep. So if you come down with the infection, that means that you've ingested those cysts from other waters. So it is one of the most common GI tract infections in all of the world. It is estimated that there are 250 million infected worldwide. Symptoms will only occur in about a third of those that are infected, so it's easy for this infection to go unnoticed. It doesn't cause acute diarrhea. It's not sudden or severe. It's chronic. So that means loose, watery stools for a long period of time that may or may not be that noticeable. The infection is in the small intestine. So what that means is there's going to be some water absorption that's inhibited, and that's what results in the diarrhea. But nutrient absorption is also inhibited. So when nutrients aren't absorbed, you can lose weight and become malnutritioned. In order to prevent the infection, drinking treated water and if you go camping and you're drinking water from rivers, make sure you bring iodine tablets or you filter your water. And because it's an anaerobic infection, it's an infection of the GI tract, and it's caused by a protozoan, we had one antibiotic that worked against anaerobic protozoan infections, and that is metronidazole. So metronidazole on our antibiotics chart was listed as an antibiotic that could be used against protozoan infections. Okay, that's all I have for you today. Thank you for listening, and I will see you again later in the week. Next up is going to be pathogens of the genitourinary tract, our last lecture of the year.